Welcome to this explainer. Today, we're decoding a truly fascinating biological puzzle. Have you ever wondered why your brain comes pre-installed with a system that perfectly responds to a plant? Way before anyone ever thought to utilize cannabis, your brain was already manufacturing its own version. A whole family of molecules released on demand to soften pain, quiet fear, blunt bad memories, and flood a running body with calm. We're going to look at exactly how this works.
So, picture this. It's 1964, and Raphael Mechoulam in Israel finally isolates THC, the main active ingredient in cannabis. But this isolation immediately sparked a massive question for neuroscience. THC is a foreign molecule from a flowering plant. So why on earth is the human brain studded with receptors that fit it so flawlessly? Since evolution doesn't build locks for keys that don't exist, this was a loud, flashing clue that our bodies must be producing a homegrown internal key.
To figure this out, we're going to map the timeline of how scientists cracked the case over several decades. We go from isolating THC in the 60s to finding the brain's biological lock in 1990, followed by the body's internal key in 92, and finally nailing down exactly how the whole system operates in 2001. Now let's look at this biological lock, known as the CB1 receptor. When Lisa Matsuda and her colleagues at the National Institute of Mental Health cloned this in 1990, they made a stunning realization. CB1 isn't some rare, specialized anomaly. It's actually one of the most widespread receptors in the entire human brain. It literally blankets the cortex, the hippocampus, the basal ganglia, and the cerebellum. A system this massive just doesn't exist purely to process a recreational plant. Being everywhere means it does something utterly essential for our survival. Fast forward to 1992, and Mechoulam's lab finally found the native key for that lock. They isolated a lipid molecule from a pig brain that bound perfectly to the CB1 receptor. They gave it a name entirely worthy of its effects, anandamide, perfectly translating from the Sanskrit word for bliss. A few years later, they found an even more abundant one called 2AG. But here's the truly wild part. Unlike normal neurotransmitters that wait around in little storage bubbles, these endocannabinoid molecules are manufactured completely on demand right out of the fatty membrane of the neuron itself, in the exact moment they're needed.
Breaking the textbook. The backward dimmer switch.
Here is where things get really crazy with a process called retrograde signaling. Normally, brain communication is a one-way street. A sender neuron shoots chemicals across the gap to a receiving neuron. Endocannabinoids completely flip that script. When a receiving neuron gets way too hyperactive and floods with calcium, it synthesizes endocannabinoids on the spot and fires them backward across the gap. They land right on those CB1 receptors sitting on the sending neuron and essentially say, "Hey, quiet down." It's literally hitting the biological brakes. This perfectly illustrates how these molecules act as the nervous system's dimmer switch, making absolutely sure no individual circuit runs too hot. Back in 2001, researchers Rachel Wilson and Roger Nicoll nailed down this exact mechanism, proving that neurons can reach backward to turn down their own incoming volume. Too much excitation in the brain causes massive damage, and too little means the system goes totally silent. This retrograde trick brilliantly solves the brain's deepest problem.
The master of balance, maintaining homeostasis. To maintain homeosis, that perfectly stable internal state no matter what's going on outside, this system operates like a master mixing engineer. It rides the faders across all sorts of critical human functions simultaneously. When a circuit runs too hot, the biological brake engages. CB1 receptors in the hypothalamus shape your appetite, which totally explains why cannabis is famous for giving people the munchies. Receptors on pain pathways turn down the volume of pain. There's even a second receptor, CB2, sitting mostly on immune cells regulating inflammation. It is a general-purpose stabilizer for your entire body.
Unlocking the runner's high. It was never endorphins.
For decades, we all credited that euphoric runner's high to endorphins, right? Well, that story was actually pretty flawed. Endorphins are huge molecules that really struggle to cross the blood-brain barrier, making it super unlikely they alter your consciousness mid-run. But a 2015 study by Johannes Fuss flipped this on its head. When mice ran on a wheel, their endocannabinoid levels skyrocketed, dropping their anxiety and pain. When researchers blocked opioid receptors, the runner's high was still there. But when they blocked the cannabinoid receptors, the calm and pain relief completely vanished. That post-run bliss is literally written in the language of our own inner cannabis.
Erasing fear and trauma, editing emotional memory.
Beyond just a hard workout, this system carries immense emotional weight. A landmark 2002 study by Giovanni Marsicano stripped mice of their CB1 receptors to see what would happen. Now, these mice could easily learn to be afraid of a tone paired with a shock, but here's the kicker. Their brains were strikingly impaired at letting that fear go once the tone became totally harmless again. Endocannabinoids in the amygdala were absolutely essential to quiet that fear circuit. Without this biological system, the brain literally cannot let go of a learned threat, placing endocannabinoids right at the very heart of trauma recovery.
And this is exactly why we're able to update old, scary memories with new, safe ones. It actively tames the amygdala when we are re-exposed to a trigger in a safe environment, making it a huge clinical target for PTSD and anxiety therapies. But editing memory is a double-edged sword. It perfectly explains why flooding your brain with heavy THC heavily impairs your ability to encode new short-term memories. The exact same molecular hand that helps release an old fear can also completely jam the formation of a new memory, messing with that delicate dimmer switch.
The blunt tool problem: psychedelics, cannabis, and caution.
Because this system is so deeply wired into memory and fear, we really have to understand the risks of interacting with it artificially. This is super critical with the rising trend of mixing cannabis with classic psychedelics like psilocybin or LSD. Classic psychedelics operate on entirely different systems, mostly serotonin. By combining them, you are forcefully nudging a brain-wide fear regulator in ways science hasn't fully mapped out yet. Because it controls our baseline for fear extinction, tossing cannabis into a psychedelic experience can wildly intensify anxiety and easily destabilize a difficult moment.
We've got to remember that using blunt tools on such a ubiquitous system carries incredibly high variability and real risks. Because CB1 sits on so many different circuits, you simply can't pull one lever without moving a dozen others. We saw exactly this when experimental weight loss drugs were designed to block the CB1 receptor to reduce appetite. Sure, they succeeded in lowering appetite, but the drugs had to be completely pulled from the market because they crashed patients' moods into severe depression and suicidality. Nudging a truly brain-wide system produces a total scattershot of unpredictable effects.
To bring it all together, the endocannabinoid system reveals this beautifully complex, quiet layer operating just beneath our daily consciousness. We don't possess a cannabis receptor just to get high. We have it because evolution required a highly intricate biological brake pedal to keep the brain perfectly tuned. It's the reason sustained effort feels like bliss, the reason we can finally let go of our fears, and a homemade calm built right into our own neural membranes.
And that leaves us with a final provocative thought to wrap up our explainer today. If your brain is operating as an incredibly sophisticated mixing engineer, constantly riding the faders of your mood, appetite, fear, and memory just to keep you in perfect balance, are we respecting that delicate dimmer switch of the self? Or are we recklessly jamming those faders with blunt external tools? It's absolutely a question worth asking the next time we think about the delicate chemistry of our own minds.